Okay, so where do stem cells come from? Where do you get these things? So think about it. If you want an undifferentiated cell that hasn't become anything yet, where would you get that? Well, you would get it from a little undeveloped baby, a little embryo. And that's where we first got them. We have figured out other ways now. But that is where we first got them. So you might be like, well, where do you get an embryo? So the first place was from IVF clinics. So there's women who freeze their eggs. They have eggs removed, and then because maybe they can't get pregnant on their own, it's not working for them. So they take the woman's egg out of her body, and they take her husband's sperm, and they get it together, and they fertilize the egg because for some reason the man and the woman just can't get his sperm to fertilize her egg. Maybe his sperm doesn't work, so they take a donor sperm. Whatever they're doing the sperm and the egg outside the body. So they have these eggs that have been like frozen and put in clinics. Okay, so let's say that woman gets pregnant, she has a few babies, she's got all these extra eggs that like can be used. So they can't just like hold on to them forever, keep them frozen for years and years. So they will eventually get destroyed. So what they can do is say, hey, lady, would you like to donate those eggs for research? Because they're just going to get destroyed anyways. So that is where we do get those eggs, is like eggs that were, going to be implanted, but there were extras that never got used. So that's where we get them. Originally, we did get them. There are some of the first that came from. So that's why there are some people who are very against stem cells, and they don't want any medicine that came from stem cells because they're against abortion. That is not done anymore. It was done a long time ago. But there are maybe some medicines now that were like made using those original cells from the abortion. So there could still be like some around that like came from abortion. So when an egg first gets fertilized, it's like a zygote. Um, you had the egg and the sperm, and now they got together and they start dividing into more cells and more cells. We call it a blastula and the gastrula. So you have all these cells that like they haven't become a brain yet, they haven't become a heart, they haven't become skin. They're just like these blank cells. So we call them stem cells because they can kind of stem to become anything. So that is where we originally got stem cells. So here's a cool little video you can play in your own time. But this is like what I just said. This is a fertilized egg kind of developing into more cells and more cells and more cells. You can kind of see the cells sort of dividing and kind of taking that shape of the gastrula. And once it gets that shape, this thing around the outside is actually going to be kind of like the babies like this. So this is like the very, very, very beginning of human life. This is going to be like the head. This is going to be the spinal cord, and does kind of start to look like a baby curled up. So that's the very beginning. That's in a zebrafish, the embryo. And that is where we originally got stem cells from, because where do you get cells that haven't become anything yet? A little tiny baby. Now we have also figured out that even apparently we still need to grow stuff, right? We still need to replace cells. So you can find stem cells all over an adult body now, in the blood, skin, urine, the pulp of the baby teeth, hair follicles, even tissue after a person has died. So the down part of this is that these are in adults, so those cells are like further along in development. It's not like when it's in a brand new little baby embryo, but you can find stem cells in there now. So we do have different types of stem cells. We have pluripotent and totipotent. So they have the potential to become anything we need. So these are like limitless stem cells. Multipotent are already kind of far along, like they've already kind of started to become bone. So you can just use them to make different types of bone cell. Or maybe they've already become like started on their way to become skin cells, so you can use them to become like different types of skin cells or different types of blood cells, whatever it is. So they have potential, but they're already kind of programmed and on their way to becoming something. And then induced pluripotent is saying, okay, well, we're going to take some of those that are already kind of on their way, like adult stem cells, and we're going to zap them and induce them and make them be pluripotent. And we've figured out how to do that, to make stem cells from an adult kind of more resemble embryonic stem cells by like zapping them and making them become like kind of bringing them back from, oh, I was almost a blood cell, kind of bringing them back to like blank and nothing, an undifferentiated stem cell. Okay, so how do you program a blank stem cell, an undifferentiated stem cell, to become what you need? So I was always confused by this. I knew you programmed them, but I had no idea how. So to answer that, we're going to look at how your body already programs its cells to be what you. So there's a lot of factors that your body uses. So we try to imitate that. There are cell signals. We talked a lot about like hormone and electrical and nerve signals in your body. Those signals tell the cell what to become. The environment that it's in, like if a cell is kind of near your heart, it's going to like get signals from the other heart cells to become a heart cell. The location, just like that I said, what cells are around it. If you put cells in the brain, it's going to be around other brain cells and it'll probably become a brain. And even these are two harder things that you don't need to know. So you don't need to know all this, but basically what I want you to know is that these are all signals, and this is all like a signal pathway that tells this cell what to become and like what genes to express or to use. So all I want you to get from this slide is that there are like a ton of signals that program a cell to become what it is today. And it does kind of follow a sequence. So let's say this up here, this is like, this is a stem cell, okay? And it's not like you just give it one signal and it becomes, what is this? It becomes a brain cell. Or it becomes, I can't read what that says, whatever that cell is. Here's a lung cell. So it's not like you can just give it a signal, one signal, and it becomes a lung cell. It's kind of like it goes through this path in its life. So first, maybe you have to give it these cells, activin A, FGF basic, nodal, Wnt3. So you give them all these signals that we've discovered by scientists, and you've got it to become like kind of this type of cell. And then you give it another bunch of signals and it becomes this cell. And then maybe you give it more signals to become a more specific cell. So it's like a whole series of signals. So basically, it's really hard for scientists. They have to figure out all these different signals, what order to get a cell to become what they want. But we are doing it. That's really hard. So I kind of say to relate this to your life, like how it's like through their life that they become who they are just by like what happened to them in their life, the cells. So it's kind of like you. Like, let's say I became a science teacher because, first I grew up with parents who valued education and took me outside a lot. And then in 7th grade, I had a teacher who said, hey, you're really good at science. So it's like throughout your life, all these things kind of push you in that direction. It's not just like one thing. So that's what's kind of hard for scientists is they have to figure out all those different things to do to a cell's life to signal it and push it in that direction to be a brain cell or something. So speaking of signals, let's just remember what we've learned all throughout is that when you are sending signals, the shapes have to match, right? So a triangle signal fits into a triangle receptor. So that's another thing scientists have to worry about. They have to make sure that the signals can be received by that particular cell because shape matters. Only certain cells can receive certain signals. And remember, the cell shape determines function. So just like I said, like where you grew up, who you're friends with, who your parents are, what part of the country, the world you grew up in, that shapes you into who you are. Same thing with cells. What they grow up around is going to determine what they are. So I said, like, if you grow up around other brain cells, you're going to receive a lot of signals to become a brain cells. So there's other things too. There's like these scaffolding fibers around the cell that are going to influence what it becomes. The neighboring cells, if you have brain cells around you, it's going to make you become a brain cell. Even the stiffness of like the liquid you're in, like if you're in a really stiff fluid, you're more likely to become a bone cell. And if you're in really soft fluid, you're more likely to become a brain cell because brain cells are soft. So when scientists are like growing these cells in a little dish, they even have to pay attention to like how hard the surface is that they put the cells on. Are they putting it on a soft surface or a hard surface? So really tricky stuff they have to get just right. Okay, so just to conclude, how do we program cells stem cells to be what we need them to be. Well, we try to do what the human body does. We try to mimic it. So the human body sends different signals and like little growth factors. So we try to figure out what those signals are and give them in the right order to become the right thing. Even like the goo, it grows up in the cellular matrix, right? It has to be like the right softness or the right hardness. So like when you're growing the cells in a little dish, like the goo that you grow it in, that has to be like the right stiffness. Even the time of day can matter. Location, the neighboring cells you put into the dish. So how do we program stem cells? We try to give them the right signals, the right goo, the right matrix that they grow up in, the right environment, the right neighboring cells. So it's tricky, but we are doing it. And lastly, here's an example of stem cells being programmed successfully. I worked at RPI a couple summers ago, and if you can see, these were stem cells. They were embryonic. They could do nothing. They were totally blank on differentiated cells. And after adding a bunch of different chemicals and signals to them, you can see they actually started to beat because we made them into like heart cells. So you can see them kind of contracting right here. We actually got them to act like heart cells, which we call cardiac cells. And then one more thing is, how do scientists like figure this out? Like, how do they know which signals work? They take like this huge tray with all these little like wells in it. See these little circles, and they put like stem cells in each of those, and then maybe in this cell, they put this signal, and this cell they put this signal, and this cell they try this liquid and see if this firmness helps. So they basically just try it in like examples, and they see which one works.